Proteins are the most structurally and functionally diverse group of biological macromolecules, and their monomer is the amino acid. And the amino acid on the right, you can see the amino group in blue, the carboxyl group in green, a central carbon, and a red variable group. While there are 23 different amino acids, we will only be talking about 20 because those are the only 20 that are biologically active. The R group is a variable group. So the 20 different biologically active amino acids are only different in the R region, and that is going to give it different properties. Some will be hydrophilic, some will be hydrophobic, some will be polar, some will be nonpolar, some will be acidic, some will be basic. But overall, the amino acid itself functions as a buffer because the carboxyl group is a weak acid and the amino group is a weak base. When you want to make a polymer of protein, you need to make a polypeptide, and you do that by taking amino acids and joining together with a peptide bond. That bond will always be created on the C terminal. That means that proteins have directionality. You have the N terminal and the C terminal. That free OH group from the carboxyl at the end is the only place you can add new amino acids. Proteins have a very complex 3D shape, as we'll see later on, and that does mean that a protein could be made up of more than one polypeptide folded together. So proteins are very structurally diverse, therefore they are very functionally diverse, and proteins are involved in almost every function in the human body. Enzymatic proteins allow chemical reactions to occur simpler by lowering the energy of activation. Structural proteins are used for support, such as connective tissue and the keratin that forms up hair and fingernails. Transport proteins move substances throughout the body, such as hemoglobin, which carries oxygen from the lungs to the other parts of the body. Some proteins function as receptors and binding, which allows cells to recognize each other. Antibodies are for defense and are made of protein. Contractile proteins like actin and myosin make up your muscles and allow you to move. Hormones, proteins coordinate body activities such as insulin, which regulates the amount of sugar in the blood, and some plants even use proteins for storage, like the proteins that make up beans. The structure of protein is very important, and if it folds incorrectly, then proteins will not be able to do their jobs. Chaperone molecules help guide protein folding by sealing the protein end during the folding process to keep it safe and allow it to fold without any disturbances. When it's folding, we find that there are 4 levels of structure. The primary structure is the amino acid sequence. So it's going to be the peptide bonds. It's going to be determined by the DNA, and very small changes to this can have major effects to everything else. The secondary structure is going to be the local folding, where you have interactions between the adjacent amino acids forming hydrogen bonds and forming one of 2 possible shapes, the alpha helix and the beta pleated sheet, as we can see below. Now, one protein can have both, but each region can only have one. The next is going to be our tertiary, or our 3rd level of structure, and that is going to be the whole molecule folding. And that's going to be guided by hydrophobic interactions, where the hydrophobic molecules fold in and the hydrophilic molecules fold out. This will be held together through a variety of bonds, including disulfide bridges, which is where the sulfur comes in a protein. The 4th level of structure does not occur in every protein, but it is where multiple tertiary structures come together to form one functional protein. Hemoglobin, for example, is made of 4 protein subunits. Overall, you can see how the structure is made. If you alter this structure, if you disrupt this structure, that's called denaturing the protein and will have major effects. There are 3 major ways to disrupt the 3D shape of a protein. If you alter the pH of the molecule, you'll disrupt the bonds by changing the H+ concentrations. If you change the temperature, you're going to increase the motion of the molecules, which is going to tear it apart. So too high temperature is going to be very bad. If you change the ion concentration, or the amount of salts, you're going to disrupt the bonds. Ionic bonds and hydrogen bonds can be torn apart and altered by the presence of charged particles. So some proteins are able to recover from this, but some cannot. Think about cooking an egg. When you cook that egg, you permanently denature those proteins. The last of the four macromolecules is the nucleic acid, whose job is to store and transmit hereditary information. The major two examples are the RNA, or the ribonucleic acid, and the DNA, or the deoxyribonucleic acid. The basic structure of nucleic acid is made of the monomer nucleotide. The nucleotide is composed of three major parts: the phosphate group, the pentose sugar, and the nitrogen base. If you look at the pentose sugar, the carbons are numbered, with the 1' carbon being bonded to the nitrogen and the 5' being bonded to the phosphate group. Now, phosphate groups provide energy during DNA replication, and they bond with the 5-carbon sugar to build the sugar phosphate backbone of DNA and RNA. But due to the location of the phosphate group, the polymer can only grow in the 5 to 3' direction, which means the new phosphate group can only form a bond on the 3' carbon, which is very important later on in the course. This does give DNA directionality, like proteins, so please make sure you understand where the 1, 2, 3, 4, 5 come from. It is just the numbering of the carbon. Now there are two types of nucleotides based off the different nitrogenous base that is bonded to the pentose sugar. The purines have a double ring nitrogenous base, the pyrimidines have a single ring base. The way I always remembered it was the longer word had the smaller structure. The purines, adenine and guanine, can be remembered as pure silver, Ag being the elemental symbol for silver. The pyrimidines are cytosine, thymine, and uracil, or cut. You have to cut the blocks of the pyramid. When you actually build the polymer of DNA, you need to form a backbone of sugar and phosphates. So the nucleotides will be bonded together from the 5' to the 3' direction using a phosphodiester bond, here represented in red, which is formed through dehydration synthesis. The nitrogenous bases hang off the sugar phosphate backbone, and what we see is that they bond across using hydrogen bonds. The two strands are complementary. A will always bind to T, and G will always bind to C. Hydrogen bonds are very, very weak, so why is this here? Well, it becomes easy to unzip the double helix, which allows replication, it allows your enzymes to read your genes to produce proteins, and so that's very important. But why does A always bind to T? Well, A to T forms 2 hydrogen bonds. G to C forms 3. You'll also notice that A and T, one is a purine and one is a pyrimidine. G and C, one is a purine, one is a pyrimidine. This keeps the width of the nucleic acid the same all the way down. There are 3 major differences between RNA and DNA. The first is the sugar. RNA has a ribose sugar, DNA has a deoxyribose sugar, but both are still 5-carbon sugars. RNA has a single nucleotide chain, and DNA has a double nucleotide chain. This does mean that DNA, like proteins, have more than one level of structure. In this case, DNA has both a primary, which is the sequence, and a secondary, which is the double helix. The last difference is that RNA uses uracil, whereas DNA uses thymine, which you can see in the image shown is not very different, but is one of the 3 major differences between DNA and RNA. Now for a quick overview of macromolecules. The first macromolecule is a carbohydrate, which we see as structure and energy components. The monomer is a monosaccharide, and the polymers depends on how many sugars you put in. The bond name is a glycosidic linkage. Lipids are long-term energy storage, hormones, components of cell membranes, and so on. They have more than one monomer, therefore they are not polymers. Fats, oils, and steroids are all examples of this, and their bond is called an ester linkage. Proteins have a wide variety of functions, including enzymes, structural components, contraction, transport, and they are made of amino acids. Their polymer is the polypeptide, lactase, hemoglobin, collagen, and their bond is the peptide bond. Nucleic acids is your information storage, and the monomer is a nucleotide. The polymer is the nucleic acid. DNA and RNA are our 2 types. The bond that holds nucleotides together down the backbone is a phosphodiester bond.